First award is going to be the John Kendrew Award. This is an award which has a long tradition. It has 20 recipients to date. It is an award which is recognizing the science which EMBL PhD students do when they leave EMBL and when they go into the world and continue doing science. And this award has been over the years very generously sponsored and supported by Roland Specker. Roland usually is here with us. Unfortunately, this time he couldn't make it due to some health issues. I don't know exactly, but basically let's just give him an applause, because without him there would be no prize. Okay, and this year's recipient, as you already know, and is actually Irma Querques. I will not say anything about her because other people will speak about her, but I have to say that she has she has amazing abilities, and one of those is that she's able to travel back in time, right? Because if you look at that little booklet which you see here, she did her PhD at EMBL from 2014 until 2010, right? So it's quite an achievement, We almost met as you were doing, this kind of. Well, actually, no, I was 10 years later. Oh, no, different generation. Sorry, I'm old. All right, good. So now, basically, we will have a special situation here because the supervisor, the PI of Irma couldn't make it, but she really wanted, to introduce her. So we will hear from her a recorded message. And that's basically what I would like to play next. So let's start.
Hello everyone, I'm Orsolya Barabas, former EMBL group leader in the Structural and Computational Biology unit. I'm delighted to learn that Irma Querques receives the 2025 John Kendrew Young Scientist Award. And I'm terribly sorry that I cannot be there with you at the EMBL Heidelberg in person today. I would have absolutely loved to share this moment with Irma and with many former and current colleagues at the EMBL. So Irma was a PhD student in my lab from 2014 to 2018. I still remember reading her first email on a late evening, totally impressed with its enthusiasm and clarity, and thinking, wow, we must have her in the lab. Throughout her PhD, Irma never stopped to impress us with her energy, curiosity, and rigor. Her perseverance was also remarkable, as she tirelessly fought through the seemingly hopeless challenge of understanding sleeping beauty transposition and finding uncharted paths to its biotechnological development. Besides research, Irma stood out as a science communicator. She is a natural in captivating her audience and happily uses unconventional methods, as crazy as hip-hop dancing, to communicate her message. She has been selected for the Kendrew Award for her work in elucidating innovative mechanisms for DNA editing, paving the way to novel opportunities in biotechnology and medicine. Her work focuses on a special class of mobile DNA elements which have a rare capacity to use RNA sequences to guide their insertion into selected genomic sites. These works now open new possibilities to build new genome editing tools that can deliver substantial DNA cargoes precisely to selected DNA locations with using a combination of transposition and CRISPR. As a dedicated science communicator, Irma passionately engages in dialogue on scientific, ethical, and societal aspects of genome editing. She has contributed to platforms like the Nature Biotechnology and Bioengineering blog and shares her knowledge with diverse audiences through TED Talks and science festivals. After EMBL, Irma was a postdoc with Martin Jinek at the University of Zurich. Currently, she is assistant professor at the University of Vienna and the group leader at the Max Perutz Labs, where she leads a young, dynamic team focused on CRISPR-associated transposons. Irma's work in society and science have been recognized by various awards, including the Brankovice Fellowship, an ERC Starting Grant, and the Valley Scholar Award. So now I wholeheartedly congratulate Irma to the Kendrew Award and wish her continued success in her future endeavors. I also wish you all a great time listening to Irma's science, which I'm a great fan of myself.
Okay, so that was great. And now I will like to invite to the podium Christoph Müller, who is going to say a few words about Irma and then help me give her the prize.
That's right. So yes, hi everybody, my name is Christoph Müller. As you heard, I've been the head of the structural and computational biology unit until last year. And I'm mainly here to set the record straight. So Irma did her PhD from 2014 to 2018, so not back in time. She was Orsolya's third PhD student, and she was absolutely great to have in the lab. Fantastic student, very pleasant to interact with, very active in a bottom-up way, working for crystallography courses, doing outside activities, and so on. I have been also part of her PhD thesis committee, which was a real pleasure, and we kept in contact afterwards because I wrote also reference letters for her jobs. I should say that was a very easy task. I had to write 3 reference letters, and she got 2 positions offered after these letters, and she accepted one, so it wasn't a big thing. Today my task is to hand over the prize because Orsolya unfortunately is not there. And so, yeah, it's my great pleasure to present you the 2025 John Kendrew Award in recognition of your groundbreaking research in genome engineering. So I guess now I have to do what?
All right, so good afternoon, everybody. It's a pleasure to be here. Thank you, Pavel and Christoph, for handing over the prize. Thanks, Orsolya, for the great introduction. I really want to thank the EMBL Alumni Association for the prize and the jury for selecting my application. And you know, for me, coming to EMBL is a bit like turning back home. And that's because at EMBL is the first time I felt a sense of community, a sense of belonging that I could experience again today at the lab day. And EMBL is also the place where I first discovered, and then I had the opportunity to follow my scientific passion for transposons, together with hip-hop dancing, obviously. And I'm particularly happy that the award celebrates this work on transposon biology because it was a rather niche research field where I began my PhD here at EMBL in Heidelberg. So I always found myself fascinated by DNA, not as a static archive of information, but as something dynamic, alive. And that's because DNA can change. DNA can rearrange, can duplicate, can mutate. And these changes are actually fundamental to determine how cells behave or how organisms evolve or how diseases develop. And therefore, in the year, I became more and more fascinated about the molecular basis behind these changes. So before EMBL, I studied biotechnology at the University of Bologna, and that's when I actually learned that most of our, more than half of our genome, including the genome of many other organisms in the tree of life, are actually made of repetitive mobile DNA sequences. And textbooks call them junk DNA, dead DNA, useless, something not to care about. But actually, this made me more curious because nature doesn't preserve for a million years something that without a reason. And in fact, when I was looking for a master's thesis position, I came across Orsolya Barabas' website at the EMBL Heidelberg. And actually, I realized that I knew nothing about transposons, but there was so much more to learn about mobile DNA. And in fact, what I learned is that actually transposons are important for genome regulation, for genome function. So for instance, in development, they are important to define some traits that make us uniquely human, for example, cognition or brain complexity. And on the other hand, transposons are also major drivers of evolutionary changes. So for instance, a transposon is behind the emergence of this black phenotype. So basically, the insertion of a transposon is important to turn a white pepper moth to black that were particularly important, conferring this advantage during the Industrial Revolution. And also, transposons are major drivers of the spread of antibiotic-resistant genes, which is a global health concern that we are still fighting today. So transposons are actually drivers of change. But the reason why I got hooked is because you can actually turn transposons into genome editing tools. And that's when I started my PhD in Orsolya's lab. I started my work on a transposon named Sleeping Beauty. So Sleeping Beauty is a transposon that has been resurrected, reawakened from a 10 million years long evolutionary sleep. And this beauty actually lies in its simplicity. So this system consists of a single protein, the Sleeping Beauty transposase, and two DNA sequences named terminal inverted repeats. And then this protein is able to recognize the terminal inverted repeats and cut any DNA framed by this sequence and paste it very efficiently in the human genome. So Sleeping Beauty, when I started my PhD, was already used in clinical trials in CAR-T therapy. And in these therapies, the immunocells are isolated from a patient, and they are genetically engineered using the Sleeping Beauty, for instance, so that they can express a chimeric antigen receptor. And this receptor makes immunocells able to recognize and fight cancer in the patient. So during my PhD, I wanted to understand how Sleeping Beauty worked at the molecular and structural level in order to understand what's the function then in the therapeutic application. However, no matter what I tried, my aim was to crystallize the protein, but we also consulted with experts from Grenoble. I did not manage to crystallize the protein, and therefore no crystals, no structure at that time. So once we were talking with Orsolya, and we thought, okay, why not to use this protein, actually turn this protein into a genetic tool? And that's when I teamed up with Cecilia Zuliani, a colleague in the Barabas lab, to develop a bit of a crazy idea. So what we did was basically to deliver directly the Sleeping Beauty transposase into human cells together with a fluorescent reporter. And I still remember the day that we realized that the cell lit up green. So it means it worked, that this protein was able to insert this gene in the human genome. But obviously, then the next test came. We wanted to use this method to produce therapeutically relevant cells. And we collaborated together with technicians actually to use this method to modify human T cells. And we realized that these genetically engineered T cells were able to completely clear out cancer in mice model. So at that point, we looked at the data and we were very happy because I said, great, we cured the mice. And obviously, for me, was a powerful reminder that actually basic research can have an important impact on medicine and therapy. However, not everybody was convinced. I still remember reviewer number 2 initially rejected our manuscript with a single sentence that sounded something like, why should we care about transposons if CRISPR exists? And I still remember this sentence because actually it missed a bit the point. So obviously, in those years, we are still in the CRISPR era, where a Cas protein, the prototype being a Cas9, can be programmed with a guide RNA to target specific loci in the human genome. And then this protein introduces double-strand breaks. However, if we want to insert a DNA at the place of double-strand break formation, actually Cas9 doesn't help. Researcher relies on cell repair, so in the DNA repair from the cell, to actually install the insertion of this large piece of DNA. And actually, DNA repair is not particularly efficient in therapeutically relevant cells, and it also struggles with long DNA payloads. Instead, the transposon, they have their own machinery. They can cut and paste DNA on their own, and they don't rely on cell DNA repair. However, at that time, it was thought that the insertion of a transposon could be only random in the genome. And for me, 2017 was a bit of a turning point, because that's when I met Martin Jinek, a former, also part of the EMBL alumni network, and previous John Kendrew Award at a mobile genome conference, so at a transposon conference here at EMBL in Heidelberg. And exactly in that year, researchers discovered a new special class of transposons in bacteria that seem to have cooperated recruit CRISPR-Cas system. So 2 years later, I decided to join Martin's lab as a postdoc to study these fascinating systems that at present they are called CRISPR-associated transposons, or CAST for short. So in this system, the CRISPR-Cas system can be programmed with a guide RNA to define a specific target in the bacterial genome. However, this system, instead of introducing a double-strand break, they recruit a transposase that can insert the DNA downstream of the target specified by the CRISPR-Cas system. So it means that CAST can perform RNA-guided DNA insertion, making them promising tools for site-specific programmable gene insertion. But these systems are not so simple as Leaping Beauty. They actually rely on different nucleic acid molecules and proteins, including an ATPase and zinc finger protein that at that time had no apparent function. And that's why, in order to study the molecular mechanism and also the structural level, the mechanism of how CAST works, I teamed up with a colleague, Michael Schmitz, in the Jinek lab to crack these systems. So we started to understand how the Cas protein could recognize the target DNA. And when we looked at the targeting complex by cryo-EM, we realized that actually in this system, the guide RNA falls into a very intricate architecture to compensate for the very small size of the Cas protein. And this is unique to this system because instead in Cas9, for example, is the protein that is bigger, and that's the one that mediates mostly the recognition. And then we went on to study the first transposon protein, the ATPase. And we were very surprised to see that this protein starts to form filament-like structures around the target DNA. And this, this filament wraps completely the double-stranded DNA, and there's no sequence specificity regarding this recognition. And we could also see that actually only in the complex of the filament, so only the filament could recruit the transposase to the target site. So at that point, we were a bit puzzled because, first of all, we had no clue about what was the role of the last protein, the zinc finger protein. And we also didn't know how the CRISPR machinery and the transposon machinery were communicating with each other. So then we put the zinc finger protein into the game, and we realized, again using cryo-electron microscopy, that this protein is absolutely essential for bridging the CRISPR module of the complex with the transposon module. However, the most interesting results came from an unexpected finding. So in the data, we saw additional density that was not corresponding to any protein that we have thought to have used for the reconstitution of these complexes. So after a few headaches, we realized that this protein is actually a ribosomal protein in a bacteria called S15. So this protein is better known for its role in assembling the ribosome, so in translation in bacteria. However, we could see this protein could stabilize the Cas machinery and could also enhance the DNA insertion activity of this system. So our results point to the fact that in this relationship between CRISPR and transposon, we forgot that this process happens in the context of the bacterial cell and actually is the host that provides a host factor to help regulating this process. But we were also, this discovery had also another implication because actually S15 was recognized as the missing component that was absolutely essential to reconstitute for the first time functional Cas machineries in human cells, which was a key stepping stone towards their development as genome editing tools. So I hope I convinced you that Cas taught us that Cas can do something that CRISPR cannot do, meaning a targeted gene insertion. And I show you that these systems are incredibly complex. So to translate them in therapies, we will need to minimize this system and develop the delivery strategy, as I showed you for Sleeping Beauty. But Cas, from what I learned from Cas is also that nature doesn't draw a strict line between a biological system, as, for example, reviewer number 2 did. And now, thinking about it, embracing this complexity and crossing boundaries is actually really what EMBL stands for. And I've been thinking a lot about the mission of EMBL to understand life from molecules to ecosystem, because this also mirrors how I started thinking about the genome. Because in the genome, when basically, in this mission, I bring it with me when I started my own lab 2 years ago at the Max Perutz Labs. So actually, the genome is a dynamic ecosystem where different genetic elements, some are static, some are mobile, can actually cooperate or can co-evolve or can even compete with each other. And I think this new idea of thinking in terms of the genome in these terms, it really will help to understand the fundamental principle of genome organization, but also help us understand and help us to develop new technologies inspired by ancient mechanisms. And I want to end with an observation or a thought. I showed to you that actually most of the progresses in my projects were not coming from testing very elegant hypotheses or, for example, for very clean, strict, following a very strict path with clean data, but they were actually coming from noticing what was surprising, what did not work or what did not fit. And then at that point, I was caring about understanding why and how. And I think I want to share a quote also from Barbara McClintock, who was the scientist who discovered transposons. And she was saying that in science, what is important is to have a feeling for the organism that could also be a feeling for biological system. And basically, the way she discovered transposons is by observing there were patterns in the maize kernels that were breaking Mendelian rules. So she was noticing when everybody saw just the noise, she was noticing this pattern, and she asked herself why. And that's why she made this revolutionary discovery that actually DNA can move. And I think that's also what EMBL taught me, that actually, progress, discoveries don't come from brilliance or intelligence, but they actually come from care. Caring about the biological system, caring for the question, and caring also for the people behind the work. So I think that's the main message that I remember from EMBL. And now I want to end thanking everybody who care, and you cared from me. So first I want to thank my scientific mentors, Orsolya and Martin. I want to thank all my colleagues at EMBL and at the University of Zurich, and including my current colleagues at the Max Perutz Labs at the University of Vienna. I want to thank my advisors, in particular Christoph, who danced with me today, and Kiran Patil, all the collaborators and the tech transfer office, and I also want to thank friends and families, and actually I have to say that a lot of EMBL alumni are also part of this cluster of people. I want to thank again the EMBL Alumni Association and the jury, and finally I want to thank my lab, was this cluster of very colorful, motivated scientists who care about mobile DNA, and they were curious and brave enough to help me to start my journey in Vienna. And thank you very much to all for your attention.